I wanted to go over this idea I have for a formal lab report and also use it as a way to introduce you to how journal articles, scholarly articles, look in the field specifically of organic chemistry. So we have a list of experiments that you can choose to do a more formal lab report on, and talking about this video itself and the different sections that you're going to put in the lab report. And I wanted to provide a sample of an article from an organic chemistry journal. And before I do that, I just wanted to mention that there's two main types of articles that you can find if you look for scholarly articles for organic chemistry. Some of them are a synthesis, how a researcher synthesized a complex structure. So if a group of researchers was the first to synthesize a natural product, a very complex molecule, they would present a paper or write up a paper with all the different steps that they used, possibly some of the major problems that they ran into while they were trying to synthesize this very complex molecule, how they solved those problems. So that's what that type of article would be like. Another type of article is if a researcher doesn't have a specific target that the person is trying to synthesize, but a specific reaction that the person is trying to improve or the group is trying to improve upon. So maybe there's a reaction that always proceeds in low yield and this group found a way to make it higher yield, or a reaction always interferes with alcohols. You can't do a reaction with an alcohol in your molecule, and they found a milder reagent or a certain set of conditions that would allow for an alcohol to be in the molecule. Or if they discovered how to make a reaction go giving only one enantiomer of a product when normally it would give a racemic mixture, that would be an improvement. So those are the types of journal articles that you can find generally for organic chemistry. Where is it? Okay. Where's my article? Oh, here we go. So here is an article, a sample article from the Journal of Organic Chemistry. So there are lots of different chemistry journals out there. This is a one dedicated to just organic chemistry articles. And this is a methodology article. This group is trying to make some cyclic esters from 1,2-diols, 1,3-diols, and alpha hydroxy acids. And they're trying to use them for X-ray analysis. So here's a little graphic scheme. The article starts with an abstract of what they did in the article, or what they're talking about, and they also have a little graphic, a figure showing what they're doing. So they're making some osmate esters with a little ligand on them. So the osmium, in addition to having oxygen, also has this tetramethyl diamine, so that's TMEDA, on part of the osmate, on the osmium on the other side, and they are using diols or alpha hydroxy acids. They're attaching this osmate to the diol or the alpha hydroxy acid, and the reason that they're doing that is to make them very crystalline. A lot of time alcohols are not very crystalline. There are some exceptions, but you might not necessarily have a crystal structure. You might have a liquid as your product if you have a diol. And if this osmate ester is attached to it, it can be a solid crystal. And they are trying to do that because an X-ray analysis is almost like being able to take a picture of a molecule. And so you would be able to have that crystal, shoot X-rays at it, compute from the results what that crystal looks like. So crystals are not wiggling around in solution. They're kind of fixed in a certain position. you can have almost a picture of that position. You could know how the molecule is twisted. You can know what configuration the stereocenter is, R or S. That's not something you can do if the molecule is in solution in a test tube. You can't tell whether a certain stereocenter is R or S. So there are a lot of advantages of being able to crystallize a compound and take an X-ray of it and understand its structure a little bit better. So that's the reason why they are doing this particular work. And so that's what this abstract is saying. The abstract part of the article is telling what kind of problem exists in science, what are they trying to solve, what is their proposed solution, and why is their way of doing things better than what previously had existed, or what was previously known. So that is, what tab are we on? Okay. So to try to mimic this type of article, your lab report, in addition to the title and the name, which you also saw on the article as well, has a title, has the names of the people that worked on it. And then you would have a very short abstract and just say what this experiment was, which experiment were you doing. Your experiment is not done to improve upon a particular reaction or to synthesize something super complex that was never done before, but since we're doing it in the context of a class, you're trying to maybe gain experience with a certain technique, with a certain reaction, be able to relate it to what you've learned about in class. So, yours could have a more pedagogical reason why the experiment is being conducted. If you would like the certain skills, the certain knowledge that this experiment was supposed to teach you and improve upon, since again, we don't have the exact kind of body of work that a normal article would be written about. We just have a one afternoon experiment that we did, or one morning experiment. Okay, so then the abstract would not really give a bunch of details and numbers, but you could make some sort of impression of how it went for you. All right, let's go back to this guy. So the article itself is talking about the previous people who had done similar work, and where does this group's work fit into what was previously being done in this area. So even though the people who are reading a Journal of Organic Chemistry article are not the general public, these are organic chemists would be reading the Journal of Organic Chemistry. It's not the kind of magazine you pick up in an airport when you're about to jump on a flight. You would have to subscribe to this type of journal. So these are organic chemists who are reading this, but organic chemistry is huge. Not every organic chemist is going to be familiar with your specific area and why you're doing what you're doing. So that's what this introduction fits in, talking about different work that's been done before. The limitations, you can see that it's talking about the limitations. Oops. So the limitations of the stuff that's been done before, how their work addresses those limitations. This background is not something that I'm asking for in your lab report, so it's not really relevant for what you are doing. There are other courses where you're supposed to find background on the reaction and stuff like that, but we're not going to do that for our assignment. All right, then, okay. Then their article also talks about what they were able to find in their work. So let me see. Let's go ahead into the specifics of this article. So this article talks about how people had discovered before that osmate esters, we've seen osmium tetroxide before in making diols from alkenes. So this is something you might be somewhat familiar with from fall semester, that osmium tetroxide is a way that you can make like a syn addition See the stereochemistry here. The two alcohols are being formed on a specific side of the alkene. And in this case, having this huge group pointing up makes the osmate ester go on the opposite side. So the osmate ester is pointing back away from us. And then they found that this was crystalline. They were trying to make diols, found that this was crystalline, and thought, oh, well, this is probably a good way to make crystal structures, verify the stereochemistry, take x-rays of them. And so this is what had been discovered before, was making osmate esters on alkenes. So in the process of doing that, when you cleave the osmate ester, you have something other than what you started with. You have a diol instead of an alkene. And so that's where this group got the idea that maybe they could start taking something that was already a diol and make an osmate ester out of that. So the limitation that they found was that a lot of times making these osmate esters was not very stereoselective. It would make a mixture, and then you would have to purify the mixture before you could get a good crystal structure. Sometimes they could not be separated, and then they could not do an X-ray analysis of them. So they talked about how one approach was to try to use something chiral on the osmate ester instead of something flat. You see this ligand over here on the osmium is flat. So using something chiral to make one enantiomer and not the other. They said how that didn't work that well. And what they decided to do was to combine a method that already exists to make diols that are enantiomerically enriched. So one enantiomer of diol is being synthesized more than the other. One is the major product. And so they're combining that methodology, making mostly enantiomerically enriched, maybe not completely enantiomerically pure diol, and then being able to make a cyclic ester of that. So here they have previous work. They have a very convenient diagram here of what was happening here, that someone else tried to make this osmate ester, and it did not crystallize because it was not separable. And they were able to form a diol with a more selective reaction and then make the osmate ester of it. And so they were able to make this compound more enantiomerically pure. So they're contrasting what had been done before with the improvements that they have made and what their work is adding to the field. And so they talk a little bit more about what specifically they did, why it's useful, and then they talk about the results a little bit more specifically, things that they tried, problems that they ran into. Let's see. It's always nice to have a chart, so here's a little list of the different reagents that they tried, the different osmate esters that they were able to make from these compounds, the melting points, the yields. So this article has a significant amount of work in it. That's why it's a lot longer than your lab report would be. You didn't do, dozens of reactions and make lots of different compounds and compare them. You did just one reaction in your lab. So let's look at your lab report. So your lab report will combine the introduction and the results and discussion sections. So the background would just be very brief. I don't want you to go and do a bunch of library research, maybe just your textbook, your notes, a little bit about what you know about the reaction. What did you learn in class about the reaction that you did in lab for the topic that you picked? So if you're doing report on the elimination reaction. Oops. So for example, if you're doing a report on the lab where we did an elimination reaction, you can say something about what you learned in class about elimination reactions. Generally, they are heated. Did you heat yours in lab? So you learned in Grignard reactions that they are not compatible with alcohols or water. Did we have alcohols or water in our Grignard reaction in the lab? So something like that. And then after that, we want a goal of the experiment, including if, drawing out the reaction in ChemDraw. You can label it Figure 1 and describe what this is. Figure 1 shows the reaction performed, a Grignard reaction on acetophenone, we did it on benzophenone. So you could draw the reaction out and describe it in the description of Figure 1. And then your results would be part of this as well, because we're not having a huge amount of background written up. So the big picture of what happened in your lab. Did you have some major problems? Did you observe anything interesting? You could also include the quantitative results at this point, some numbers, the percent yield, the mass of the product that you obtained, the analysis that you did. How do you know that you had the correct product and it was pure? So similar things to your worksheets, but just typed up a little bit more. And then also, don't forget that these are done in a passive voice. So, refer back to the sample article. You can see that the type of sentence is usually a passive voice sentence. So several other bitentate structures were evaluated under standard conditions. So they don't say that we evaluated, or the head of our group evaluated, or, Sarah evaluated. They just kind of say something was done, and they don't say, we were confused. They say it was not clear to us whether they were stable, or protic solvents tended to result in no reaction. So you could talk about objects as being the subject of a sentence. So you could talk about what the chemistry did, what the solvents did, what the heating did, but not what specific people did. So it's kind of the convention of how science articles are written. And then a little bit of what you learned, again, done in a passive voice, about the experience that, what the experience taught you. But just instead of saying, I learned this, you could just say that, this part was easy, this part was difficult. More rigorous washing was needed because the product was not pure of side product biphenyl compound, something like that. And then a conclusion section. Let's go back to the article and see the conclusion. Where is it? So after describing a lot of the work that was done in this article, there is a conclusion section, very short summary, so that in case your attention starts wandering during the article, you can figure out what the most important things that you should have gotten from this article were. After the conclusion, that's where they include an experimental section. The actual procedure is here. Let's see if I can move this over. So these are the details of what was done. A solution of TMEDA, this many grams, this many millimoles in acetone. So these are the actual specifics. How concentrated was the solution? What scale was it done on? What was the volume? What happened? A white precipitate formed. The mixture was cooled. The solution was filtered. They don't have in this particular article a copy of the NMR. I'm kind of surprised that they didn't put the melting point, since it was a solid, but I guess that's in one of the, some of the charts have the melting point instead. So they have a list of the proton NMR. It was done on a 600 megahertz machine. The solvent was deuterated methanol, and these are the shifts. They had something at 7.72 ppm. It was a doublet and the splitting, the distance of the splitting was 8.3 hertz, it integrated for 2 protons. We had something at 7.26, so et cetera, et cetera. Carbon NMR, they have the data for that. Also, so now that we have the internet, years ago we didn't have more than just a printed copy of the journal, but since a lot of this stuff is online, there are more copies of the spectra. If you really wanted more details, you could go online and search out some of the some additional information if you wanted. And then this is how to get in touch with the author if you want to try this for yourself and you're running into problems. These are the footnotes for the references that they used during, the write-up. They had they had some footnotes for where to find the other articles that they referred to. Oh, this was done before, this was not able to be separated. So they have references to those papers that they put in the article. So you don't need references in your specific assignment. I just wanted to point that out. But we do have a procedure section for you to do. Let's see, what do we have? So short experimental, because since you do have a procedure, you know what volume you used, you know what mass you used. So you would just give a short passive voice description of what was done. You'll notice that the description was relatively brief. So they would put that it was extracted, that it was filtered. They didn't put, oh, well, I set up a filter flask, put filter paper on it. You don't need all that. You can assume that whoever is trying to reproduce your work knows how to do a filtration, knows how to do an extraction, knows how to take a melting point. So you don't have to give the very detailed step-by-step that perhaps you wrote for yourself when you were writing up your lab procedure. And then what product you obtained, your mass, whether it was white crystals, tan powder, whatever, and percent yield of the product. And then the interpretation of why the analytical data showed you the product is pure would be done more in the body, in the summary of the article. This experimental procedure just has a list of the HNMR peaks. If you do a TLC, sometimes people do TLCs and include that data in the experimental. The article I showed you did not have that, but that is something that can be included. IR, the main things would be included, not just the whole entire forest of IR peaks that you have. Probably just alcohol stretch, CH stretches, whether they were above or below 3,000 is important. If there was a CO stretch, if there was a benzene ring, you might want to include the CC stretch, but not a ton of IR data. The HNMR, hopefully you know what each peak is, so you should include all of them like the article does. And yeah, so hopefully we'll see if this is a doable assignment. Like I say in the instructions, I'm hoping that this assignment is not a huge deal. Sometimes this is hard for me to judge, so let me know if it's taking you more than a couple of hour or two. Hopefully it wouldn't, because it is only 10 points. So we'll work on this assignment, see if we need to modify it at all. But that is sort of what I had in mind. Hopefully it makes sense.